Okay, good afternoon, everyone. It's nice to see you again in the virtual world, and I hope that you're doing okay. At least we have a little bit of sunshine outside the window today, so that's always encouraging. Okay, lots to do as always, but first first let me ask, can you hear me okay through the video? Please type yes into the chat window. All right, thank you very much. Okay. So folks, here we are. We are literally 2 weeks away from your final exam, okay? As I've indicated in previous emails, your final exam will in fact be Tuesday, May 12th. Tuesday, May 12th is your final exam. Okay, so please expect that this afternoon along with the normal email information that I send. I will also send to you your study guide for the final exam. Okay, so it will include things that we haven't yet talked about, but which hopefully we will talk about before the end of our 2-week period together. Okay, so before we start new content, do any of you have any questions about, end of semester activities or anything like that? Okay, all right. All right, folks. So since I'm not seeing any questions, let's go to the Microsoft Word document, and we'll start the tryptophan operon. Do you have any questions about the lactose operon from last time? I took nearly the entire class period to talk about the lactose operon. And that's because I really do want you to have a deep understanding of that system. Do you have any questions about that? Okay, everybody's silent, so I hope that means everybody's okay. Let's go and get started with the tryptophan operon. I won't have the luxury of spending the whole class period on it because we've got to get to the next chapter, but at least we can talk about the tryptophan operon for maybe 20 to 30 minutes here. Okay, so remember, we're in chapter 12. We're talking about these operons, these groups of genes that are typically transcribed from a single promoter. We covered the lactose operon last time, which you recall is in fact an inducible operon. And now we're going to give you a contrasting system known as the tryptophan operon, which is a repressible operon. Okay, so on my screen, you should now see a Microsoft Word document. Please let me know if you can't see it or if you can't hear me, just type into the chat window. Okay, so we're going to talk about understanding the tryptophan operon. Okay. We recall that the purpose of the lactose operon is to allow cells like bacteria to metabolize the milk sugar lactose and use it as a fuel supply to manufacture ATP. Well, the purpose of the tryptophan operon, as you've probably been studying on your own time, is to regulate the synthesis of the amino acid we call tryptophan. Okay. The purpose of the tryptophan operon is to regulate the synthesis of the tryptophan amino acid. Okay. So we're going to be talking a little bit, I'm not going to go into as much detail here as I did with the lac operon, but I'm going to be going into some detail about the tryptophan operon. And the first thing I want to do is make some points of contrast to the lactose operon, because I'm assuming that you kind of have that foundation of how to think about gene regulation. And I want to give you some points of contrast to bounce off of that. 2. points of contrast to the lactose operon. All right, so we'll start with the thing that I just mentioned. While the lactose operon is inducible by the presence of lactose, okay, the trip operon, okay, short for tryptophan operon, is repressed by the presence of excess tryptophan. Okay. So in the lactose system, the presence of the milk sugar actually turns on the genes to metabolize the milk sugar. But in the tryptophan operon, which the genes are typically always on, if we have an excess of tryptophan available from the environment, we don't need any additional tryptophan to get made. Then tryptophan is the very thing that helps turn the genes of the tryptophan operon off. Okay? All right, once again, the lactose operon is inducible by the presence of lactose. The trip operon is repressed by the presence of excess tryptophan in the cytoplasm of a bacterial cell. Okay? Gang, does that make sense? Are you okay right now? Okay, very good. Let me know if you have questions. All right. Let me give you a little bit more of a breakdown of here the things we are talking about. In the lactose operon, lactose binds to the lac repressor protein, preventing it from binding to the lac operator region. Okay, you remember that. The lactose binds to the lac repressor, changes the shape of the lac repressor, causes it to fall off the DNA site that we call the operator. Okay. In the trip operon, tryptophan binds to the trip repressor, changing its shape so that it can bind to the trip operator. Okay, in the tryptophan operon, TRYP for short, tryptophan amino acid binds to the trip repressor protein, changes its shape, and allows it to bind to the trip operator. Okay, so in the lactose system, lactose pulls the regulatory protein off the DNA, but in the trip operon, we do the exact opposite. The presence of the tryptophan amino acid allows the repressor protein to attach, to specifically shut off gene expression. Okay. Also, in the lactose operon, I mentioned that there is this thing called a leader, and we said that at least in the lactose operon system, the leader sequence has no really known biological function. It kind of doesn't do much of anything. It might, maybe it does do something that we're not aware of, or maybe it's simply an evolutionary fossil that's just kind of there and doesn't do much of anything. You know, we could take bets on either side what that might be. But the lactose operon has a small leader with no obvious regulatory capacity, but the trip operon has a leader that regulates gene expression in a process known as attenuation. Okay, so attenuation is something that you're going to want to spend some of your own study time on. We will talk about it here, but I'm just pointing out that it is something that you should be aware of. Okay. All right. Okay, gang. So let's go ahead and just create a very basic operon schematic in your notes at this time. And I think I'll stick with the Word document rather than trying to get fancy with PowerPoint at this moment. Okay, so schematic of the trip operon. Okay, all right, so we typically have P for promoter. Then we have trip R, and then we have two vertical arrows that are slanted, again indicating that it is physically distant from the next sequence I'm going to put into the notes. And we have a new promoter, a new operator, a new leader, an A site for the attenuator. We'll document all of these. And then we have a variety of structural genes. We have trip E, trip D, trip C, trip B, and trip A. Okay, so let's document what all of these things actually are. Okay. Okay, so question coming into the chat window at this time. How many operons are indicated on this schematic? Okay, Ian says 2. Do the rest of you agree with him? Okay, all right, we do have some disagreement. Well, again, the answer is in fact 2, and it's because we have 2 promoters here, right? We have this promoter here, which I'm currently highlighting in gray, and then we have this promoter here, which I'm also highlighting in gray. So on this drawing, on this schematic, we actually have 2 operons, okay? The first operon is the trip repressor protein operon. Okay, the first operon is the trip repressor protein operon. Let's see here. Maybe I don't know if I can use any fancy coloring here to, yeah, okay, so everything in yellow at that point is the first operon. That's the trip repressor protein operon, okay? The tryptophan operon is everything that I'll momentarily put in, well, let's see here. Let's put it in, that's not what I want. Let's put it in red, okay? All right, so everything in yellow is the trip repressor protein operon, okay? So P, of course, is the promoter, and trip R is the gene that codes for the repressor protein, okay? This gene is constitutive. It is constitutively expressed. Okay, it is always on like a leaky faucet. You cannot shut it down. Mother Nature doesn't want you to shut it down. Okay. All right, the second operon is the tryptophan operon itself, which is basically the topic of our conversation here. Okay, you see schematics of the tryptophan operon in various circumstances on page 347, figure 12 of your textbook. The second operon is the tryptophan operon in red. Okay. And let's define these things. We have a promoter. We have O for an operator. Okay, this is the binding target. This is the binding target for the for the TRP repressor protein. When the TRP repressor protein is complexed with tryptophan, okay? So the operator is the binding target for the TRP repressor protein, but not the naked TRP repressor protein, only the TRP repressor protein that is actually complexed, bound together with the tryptophan amino acid. That would be analogous to what circumstance that we talked about in the lactose operon. Can you tell me that, gang? Type that into the chat window. If we need something to bind to the TRP repressor protein to bind to the DNA, what other circumstance in the lactose operon required two things to come together to bind to the DNA? Am I making sense? Okay. What two things had to come together in order to attach, okay, to DNA? Okay. Yes, cap cyclic AMP. That's what I was driving at. Okay. All right. So we have a similar circumstance here in the tryptophan operon, that for the repressor, you need tryptophan amino acid to attach to the DNA. The analogous idea from the lactose operon is that you needed cyclic AMP to bind to the catabolite activated protein to bind to DNA. Okay, just for completeness of your notes, I think you already know the promoter is the binding target for RNA polymerase. That's always true, right? That's always true. Okay, let's continue here. We have L, which is the leader. Okay. This is a sequence of DNA that is transcribed into RNA and then translated. Okay. Notice that is different than what we said the leader was for the lactose operon. In the lactose operon, the leader is a sequence of DNA that's transcribed into RNA, but it is not translated. In the lac operon, the leader is not translated. But in the tryptophan operon, it is translated. Okay, the leader is a sequence of DNA that's transcribed into RNA and then translated. This translation is part of the regulatory process called attenuation. And we'll talk about that in just a moment. Okay. Now, in the schematic up here, we have an A drawn. Okay. Please know that the A, it's just a schematic. It's just a cartoon. The A is part of the leader. The attenuator, the A is for attenuator. And what we're saying is that there is a subset of the leader sequence which supports the attenuation process. So the A is short for attenuator, and the attenuator is a subset of the leader. Okay, this is a sequence within the leader that supports attenuation. Okay. Now, the next 5 genes, I will put some things down into your notes, but frankly, it's not that big of a deal that you know what their biochemical description is, because we're not talking about the biochemistry of making tryptophan at this moment. We're talking about regulating the expression of genes to make enzymes to synthesize the tryptophan amino acid. So we're not talking about biochemistry, we're talking about the genetics. So I'll just do a quick summary of what these things are and say, okay, if you want to know more, go do some research on it. TrpE is the gene for an enzyme called anthranilate synthase. Okay, so this enzyme synthesizes anthranilate, okay, which is the precursor molecule to tryptophan. So if you take a biochemistry course down the road in your future, you may very well talk about the biochemistry of the TrpE enzyme, but you won't focus necessarily on the gene regulation of it. Okay, so you get different things in different courses. Okay, TrpD codes for a polypeptide, which is also part of the anthranilate synthase enzyme. So what I'm telling you is TrpE and TrpD together are multiple polypeptides that come together to form the anthranilate synthase enzyme, because the anthranilate synthase enzyme has quaternary structure. Okay, so TrpD is a gene for another polypeptide that is also part of the anthranilate synthase enzyme. TrpC codes for a fancy enzyme with a big name, glycerol phosphate synthetase. Okay, and this is simply an enzyme that performs an isomerization reaction where it takes the product that's made from the activity of the TrpE and TrpD polypeptides and moves some of the atoms around to form a precursor that's needed then to subsequently build the final tryptophan amino acid. Okay. And then we have TrpA. We have actually TrpB and A. I'm going to put these, well, I'll put them on separate lines. TrpB is a gene for a subunit of tryptophan synthetase, and TrpA is a gene for a different subunit of tryptophan synthetase. Okay, so the polypeptide of the TrpB gene and the polypeptide of the TrpA gene, they come together to build the actual tryptophan synthetase enzyme, which collectively is responsible to adding a serine residue to the product from TrpZ to actually synthesize the final product of the pathway, which is tryptophan. Okay, so we'll just say here, TrpB and TrpA work together to build tryptophan. Okay. All right, gang. So, any questions at this time? Any question at this time? Okay. All right. So, let's put into the notes a chart, a 2-column chart, where I'm going to make some more comparisons between the lactose operon and the tryptophan operon. Okay. Let's see here. I think I'm going to need about 3 rows. Okay. So the lactose operon is controlled by colon. And on the right-hand side, the tryptophan is controlled by okay. So we're going to say some things here that you already know, but when you get down into the weeds of things, sometimes this stuff gets confusing, even if you've been covering it for a while, okay? So the lactose operon, as you know from last time, is regulated by catabolite repression, okay? Where the absence of glucose stimulates cyclic AMP production so that cyclic AMP can bind to the cap protein, and the cyclic AMP cap protein together can attach to the operator, can attach to the promoter to facilitate RNA polymerase binding, okay? The second thing that the lactose operon is regulated by is the lactose repressor protein. Okay. Lactose repressor protein is constitutively made. Okay, so it's always on, it's always made, and it naturally binds the operator. To block any forward motion of RNA polymerase. The only way you get the lactose repressor off the DNA is to bind lactose to the repressor, change its shape to have it fall off the DNA. Removing the repressor requires lactose to bind to the lac repressor. Okay, so all of that was covered last time. Now with the tryptophan operon, we have 2 forms of regulation, but they're not exactly the same thing. Okay, we have presence of the tryptophan repressor. Okay, this is also constitutive. Okay. In the lactose system, the presence of the lactose repressor always turns off the operon, but in the tryptophan operon, it's different because the tryptophan operon is a repressible operon. It's on until you turn it off, okay? So in this scenario, the repressor cannot bind to the operator without tryptophan, okay? If tryptophan is present, this tells the cell that there is no need to manufacture enzymes capable of making more tryptophan. Okay, let me read that again. If tryptophan is present in the environment and therefore in the cytoplasm of the cell because it's being brought in, if tryptophan is present, that tells the cell there's no need to manufacture additional enzymes capable of making more tryptophan. So in such a case, the excess tryptophan binds to the TRP repressor, changing its shape so that the tryptophan TRP repressor combination can bind to the operator and block RNA polymerase access to the downstream TRP, E, D, C, B, and A genes. Okay. Let me read that to make sure I've typed it properly. The tryptophan repressor is constitutively made. It cannot bind to the operator without tryptophan. If tryptophan is present, it tells the cells that there is no need to manufacture enzymes capable of making more tryptophan because we already have it in the environment. The excess tryptophan binds to the TRP repressor, changes its shape so that the tryptophan-TRP repressor combination can bind to the operator and block RNA polymerase access to the downstream structural genes. Okay? Any questions so far? Okay, let's keep going, gang. All right. The second part of this is the attenuation. Okay, now your book talks about attenuation on page 348, and it has a drawing there that's much better than I could realistically do. So I hope you have your textbook available. I'm looking at figure 13 on page 348, figure 13, page 348, and the title of the figure is The Attenuation Model Regulating the Tryptophan Operon. Okay, as your book indicates, that even when the tryptophan TRP repressor protein is actually bound to the operator and should be stopping gene expression, there's still a low level of expression that was measured in the laboratory. And what scientists realized that there's an additional regulatory step on top of the tryptophan repressor that's at play here. And this is the attenuation that we're talking about. So number 2 is attenuation. This is a process that further modulates gene expression even when the TRP repressor is bound to the operator. Okay. Okay. All right, so what we need to do is I'm hoping that the part of the tryptophan repressor, number 1, is fairly understandable to you because it's simply the opposite logic of what we talked about with the lactose operon. So before I go and talk about the attenuation process, do you have any questions about the logic of everything I'm shading in gray at this point? Okay, not hearing anything, so I hope that means you're in good shape. So let's talk about the attenuation business here of figure 13. Okay, attenuation of the TRP operon utilizes RNA secondary structure to accomplish the regulation. Okay, so let's say this. Okay, when RNA polymerase, okay, RNA-P is RNA polymerase. When RNA polymerase is able to access the TRP promoter, it begins making messenger RNA. Okay, it begins actually transcribing the messenger RNA. Okay, the stretch of DNA in between the TRP promoter and the first structural gene, TRPE, you know, is the leader sequence, which contains this area that we're calling the attenuator. Okay, so this is blown up for you. It's expanded, it's magnified for you on picture A of figure 13 here. Okay, the stretch of DNA in between the TRP promoter and the TRP E gene is referred to as the leader, which contains a special sequence called the attenuator. Okay? All right. Let's continue here. As that leader sequence is transcribed, the messenger RNA has the ability to fold into two different shapes. Okay, if you look at figure 13, you'll notice there's one shape there that's described as the anti-terminator hairpin, and there's the bottom shape there which is described as the terminator hairpin. Okay, so based on circumstances that are true or false inside the cytoplasm of the cell, one of those two alternative shapes or conformations are going to be formed by the leader RNA as it is manufactured. Okay, as the leader RNA is transcribed, okay, as the leader RNA is manufactured by RNA polymerase, the messenger RNA has the ability to fold. Now, when I say fold, I'm thinking about, what kind of shapes can you make with your shoestring, right? You can make bows out of it. You can make knots out of it, okay? You take a string and you can do all kinds of fancy folding and twisting on it. Well, DNA and RNA are like that. They have the same physical characteristics. So you can fold the messenger RNA into alternative structures. The messenger RNA has the ability to fold, kind of like a shoestring, okay? Into two possible shapes. Obviously, the better word for science is conformations. All right, so when charged tryptophan tRNA is unavailable, meaning that the tryptophan levels in the cell are actually low, then it makes sense, if the tryptophan levels are low, it makes sense that we want to express genes E, D, C, B, and A. So in such a circumstance, the RNA is going to form the anti-terminator hairpin. Okay? When charged tRNA for tryptophan is unavailable, okay, meaning tryptophan levels are low in the cell, the anti-terminator hairpin forms, okay? When the anti-terminator hairpin forms, because, all right, let me ask you a question to see if you're following along, if you understand your own study time, okay? And honestly, you can kind of look at the figure and figure it out, okay? What is causing the anti-terminator hairpin to actually form? What is causing the anti-terminator hairpin to actually form? Okay, John, a ribosome. I like that answer. That can get us to home plate. In other words, remember, we're talking about gene regulation in a prokaryotic cell where transcription and translation are coupled. That means before the messenger RNA is finished being manufactured, a ribosome can immediately start translating it, right? So you don't have to have the full messenger RNA made before a ribosome can hop on there and start translating it. So what happens is this leader RNA is manufactured, RNA polymerase continues to move along making RNA, and bacterial ribosomes sit down on the leader and they start to scan along to see if they can translate anything. Well, as it turns out, there's not enough tryptophan available in the cytoplasm. So that means there's no charged tRNA available. Because of that, the ribosome actually stalls. And it's the stalling of the ribosome motion which causes the RNA to adopt the anti-terminator shape. Okay? Does that make sense? It's actually the stalling of the ribosome which promotes formation of the anti-terminator hairpin. It's the slowing down, the stalling of the ribosome motion as it's moving along the RNA that causes the anti-terminator to form. Okay, so let me see where I am with our notes here. When the charged tRNA of tryptophan is unavailable, the anti-terminator hairpin forms because the ribosome has stalled its motion. Okay, because the ribosome has stalled its motion. Again, why did it stall its motion? Because there's no charged tRNAs with tryptophan available because levels are low in the cell. Okay. And then we have the opposite side of the coin, or the other side of the coin. When charged tRNA is available, the ribosome does not stall. It does not stall along the RNA, and because it does not stall, it drives the folding process, which itself is complicated and not completely understood. It drives the folding process of the RNA into an alternative shape, which you see as the terminator hairpin. If you look on figure 13, notice that your book has color-coded different areas, 1, 2, 3, and 4, red, blue, green, and yellow. And notice for the anti-terminator hairpin to form, you're actually going to have base pairing, hydrogen bonding between sections 2 and 3. But for the terminator hairpin to form, you have hydrogen bonding happen between a section in between 3 and then section 4. Okay? So when charged tRNA, TRP, is available, meaning tryptophan levels are high in the cell, the ribosome does not stall, and this promotes formation of the terminator hairpin. Okay. Obviously, if the terminator hairpin forms, you've created a shape that prevents manufacturing of the proteins from genes E, D, C, B, and A. So you shut production down of these enzymes. Okay, so this leads to turning gene expression off, whereas up above, if we have the anti-terminator hairpin, this supports gene expression. Okay. All right, so I know there's a lot there, and I've already spent more time on this than I want to, but one more question coming through here. Would this attenuation scheme work in a eukaryotic cell? Okay, thank you, John. That's exactly what I was fishing for. Okay, it wouldn't, right? This truly is a bacterial, or I shall say, prokaryotic specific gene regulation strategy. This strategy is not going to work in a eukaryote because of the presence of the nuclear membrane. That means we have decoupled transcription and translation. So this is only going to work in a bacterial system. Okay. Gang, let me show you a video on this to see if it can tidy things up for you. And we'll field any questions you might have. And assuming you have no questions, we will move on to the next chapter. Okay, so one moment while I get a video queued up here. Okay, this is the trip operon. I'll send you a link for this in this afternoon's email. And here we go.
Tryptophan operon, TRIP operon for short, is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The TRIP operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRIPR, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the TRIP operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRIPL, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRIP synthesis pathway, TRIPE and D for anthranilate synthase, TRIPC for indole glycerol phosphate synthase, and TRIPB and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRIP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the LAC repressor, the TRIP repressor does not stick like glue, but periodically dissociates, allowing a low, basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter 2 tryptophan codons in a region called 1. They wait for TRIP-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the TRIP tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
Okay, gang. So let me ask you. I don't want to beat a dead horse. Do you have any questions about this process? Anything that's confusing you? Anything I can help clarify? Okay. Let me give one more question through the chat window. Okay. True or false? The leader sequence of both the lactose and tryptophan operons are translated. Okay, very good. Very, very good. That is in fact false. I just want to be sure that you're really appreciating the difference between these two systems. Okay. All right, gang, one more time. Any questions you'd like to ask? Okay. All right, then let's get on to chapter 13. Chapter 13 is eukaryotic gene regulation. Okay. So, all right. That begins basically on page 359 and 360 of your current textbook. Okay. All right, so some opening comments here. All right. The organization of the genetic material, the information content of the eukaryotic DNA does tend to be more complicated than in prokaryotes. I mean, I don't think that's too surprising. Okay. Okay, obviously never underestimate our simpler friends. I mean, look what a simple virus is doing to the planet at this time. So we don't want to underestimate these things. But in terms of information content, eukaryotic systems tend to be richer in how the information is organized. Okay. While prokaryotic systems frequently rely upon operon regulation strategies, eukaryotes do this less frequently. Okay. While prokaryotic systems frequently rely upon operon regulation strategies, eukaryotes do this less frequently. There are exceptions to this, however. Some eukaryotic operons have been documented. Okay. One such example of the eukaryotic operon, or at least an operon similar, an operon-like structure in eukaryotes, even in our own cells, human animal cells, involves the coordinated regulation of the major histocompatibility complex genes that are very important to our immune system. One such example of an exception is the organization of human, meaning animal, major histocompatibility complex genes, which are organized in an operon similar fashion. Okay, so just lets you to know that there are circumstances that we can find organization of genes that resemble the operons that we just studied in chapter 12. Okay, number 2. Let me see here. Okay, number 2, regulation of gene expression in eukaryotes is more involved because there's more steps that you can regulate the process at, or at least there seems to be more steps that Mother Nature has taken advantage of. Okay, regulation of gene expression in eukaryotes tends to be more fine-grained, okay, kind of more tightly controlled than prokaryotes. Okay. there are multiple steps that regulation has been documented in that include, okay, so here I'm drawing upon figure 1, page 360. Okay, so we have chromatin remodeling. Okay, we've talked about that. Okay, we talked about the significance of remodeling the chromatin material multiple times this semester. Okay, so this often involves epigenetic tags that lead to reorganization of the DNA. We have transcription level of regulation. Okay, this can transcriptionally, genes can be regulated at both the transcription start point, initiation, during the actual manufacturing, which is elongation, and during the termination phase of it. Expression of genes can be regulated by transcription initiation, elongation, and termination. Okay. Now, not all genes are regulated the same way. Different genes are regulated different ways, okay? But we're saying collectively across eukaryotic cells on the planet, these modes of gene regulation have been documented by science. Okay, we also can regulate genes at the level of gene splicing and processing of the RNA. Okay, this includes physically cutting out the introns and gluing the exons together. Okay, splicing exons together. It also includes the addition of the 5' cap to the RNA, as well as the addition of the poly A tail at the 3' end. Okay, these are things that we've touched on, you know about, you've been thinking about these during homework problems. Gene regulation can be regulated at the level of transport, that is transporting the RNA between the nucleus and the cytoplasm. Okay, that can't occur in prokaryotes because there is no nucleus, but in eukaryotes that can definitely occur. RNA transport out of the nucleus can be regulated. The process of messenger RNA stability or its degradation can be regulated. Okay. I saw some recent research suggesting that the breakdown of RNA in cells can actually be used, the breakdown products of the messenger RNA can themselves also feed back to turn other genes off. Kind of a fascinating possibility. Genes can be regulated at the level of translation. Okay, this also can occur at the initiation, elongation, and termination phases. Is controlled is controlled at the initiation, elongation, and termination phases. Okay. Translation, there's a special level of translation regulation that I'm going to introduce you to in this chapter, which is called RNA-induced gene silencing, that plays a role here. Okay, we will talk about that. And then also about protein modifications. We haven't talked about many of them here, but we know from our reading that we've documented, science has documented over 200 different forms of protein modifications that can control their stability and how the cells can actually use the proteins. Okay, over 200 known modifications have been documented. All right, so suffice it to say, gang, there are a whole bunch of steps that Mother Nature has exploited throughout evolutionary history to regulate genes in a variety of circumstances, okay? And one is not necessarily more important than the other, it's just the particular solution that Mother Nature has stumbled upon at any given moment of time. Okay, any questions so far? Okay. All right, so what I want to do next is go over an example of a typical eukaryotic gene architecture. So for this, I'm going to rely on figure 6 on page 366. An example of a eukaryotic, not a, example of eukaryotic gene architecture. And for this, we're going to talk about the human metallothionein 2A gene. Okay, the human metallothionein 2A gene. All right. Okay, so well, let me describe what this gene does for you first. Okay. So we're looking at figure 6, and we're going to put this in your notes. The human metallothionein 2A gene codes for a protein that has the ability to bind heavy metals. Okay, now by heavy metals, I'm talking about things like zinc, cadmium, and others, okay, that often are damaging to a cell. Okay, because the presence of these metals actually chemically interact with oxygen in the atmosphere, or oxygen that's floating in the cytoplasm, causing the production of toxic free radicals, which then go around and smash up other molecules inside the cell and damage chemical bonds. Okay. Heavy metals are known to interact with oxygen to form damaging free radicals. Okay, so long story short, the human metallothionein 2A gene codes for a protein that sequesters these heavy metals so they do not interact with oxygen, so we do not form free radicals, so we do not damage the molecules inside the cell. So it's a way of keeping the cell safe from the potentially damaging effects of oxidative stress. Okay. So let's take a look at the basic schematic here of this typical eukaryotic gene, okay? And we're not going to get too fancy here, but we have the GRE element, okay? Then we have some things called ARE, and in this gene, there's actually 3 of them side by side. Then we have an MRE element, followed by another MRE element. I'm going to define all of these things for you, followed by a BLE element, followed by another MRE element, followed by a GC element, followed by another MRE element, followed by a TATA element, followed by the INR element, which is short for initiator. Okay, so let's define these things. All right. All right. The GRE elements are short for glucocorticoid response elements. Okay, so the glucocorticoid response elements are DNA words that allow the glucocorticoid receptor proteins involved in glucocorticoid metabolism to bind and stimulate the expression of this gene. Okay, so these are DNA words that are binding targets for proteins called glucocorticoid receptor proteins. Okay, just, I know some of you have a medical background. Remember, glucocorticoids are just a type of steroid, so they're hormones, right? These are hormones. Okay, so glucocorticoid, let's put up here a steroid hormone. Okay, glucocorticoid, which is a steroid hormone response element. These are DNA words that are binding targets for proteins called glucocorticoid receptor proteins, which stimulate expression of this gene. In other words, of the human metallothionein 2A gene. When these glucocorticoid receptor proteins bind to the GRE DNA words of this gene's expression is turned up. It's turned on. Okay. All right. So please stop me, ask questions in the chat window. There's a lot of information here to get into your notes. I will say additionally here that many genes in eukaryotic cells, including our own, many genes in eukaryotic cells have these GRE elements, meaning that steroids are allowed to influence the expression of genes that regulate a variety of processes, such as development of our bodies, glucose metabolism itself. So it plays a role in sugar metabolism, and these GRE elements have also been located in cells that are heavily involved in body defense. So I'm talking about the immune system. Okay, so we're not going to get to the end of this story here in our course, gang, right? I mean, you never get to the end of it. There's always another level of complexity that we can dig into. But for right now, be aware that many eukaryotic genes do have these hormone responsive DNA words. Okay. Next, we have the ARE segments. ARE stands for AP factor response element. Okay. AP factor response element. Okay. This is a DNA word that allows activating proteins which go by the name of AP1, AP2, and AP3 to physically attach to those locations and stimulate transcription of this gene. Okay. This is a DNA word that is a binding target for activator proteins called AP1, AP2, and AP3. Which bind to these elements, which bind to this element. And activate transcription of this gene. Okay. Now, AP1 proteins, the proteins that bind to these DNA words, they are known to be involved in a lot of cellular processes, including the regulation of apoptosis, so cell death, and tissue regeneration. So when you fall down and skin your knee outside, you can bet that AP1 proteins are binding to the genes of many of our genes to regulate tissue regeneration. Sorry about the telephone, folks. Many genes have binding targets for AP proteins to regulate apoptosis and tissue regeneration. Okay. Next, we have the MRE elements. Okay, the MRE elements are short for metal response elements. Okay, the metal response elements, these are DNA words that are binding targets for the MTF1 family of proteins. MTF1 stands for metal regulatory transcription factor 1. Okay, this is a DNA word that is a binding target for MTF1, metal regulatory transcription factor 1. Okay. When MTF1 binds, it also stimulates transcription of this gene. Okay? So if you're starting to feel like, whoa, this is kind of insane how many factors are involved and players are involved, that's really the whole point of this conversation, is that there are many, many things involved here in regulating and coordinating the expression of a single eukaryotic gene. Okay. So when MTF1 binds to the gene, it also stimulates transcription of the gene. But let me back up and say, I'll add on here, okay? We have a scenario happening here that we also have had seen in chapter 12. The MTF1 protein is only able to bind to the gene when the protein itself is actually bound by the heavy metal, okay? So when the heavy metal is present, it binds to this protein. And when the heavy metal and protein complex binds to this gene at the MRE element, we stimulate transcription of this gene, okay? It actually requires, okay, MTF1 requires attachment of a heavy metal ion to actually attach to this gene and increase its output. Okay. All right. Boy. Okay. Folks, if you can give me 5 minutes, I really appreciate it. I want to see what I can get through here. Okay. All right. Letter D is BLE. Okay, BLE is short for a basal element. Basal element, this is a DNA word that is a binding target for additional activating proteins some of which we don't know yet. We don't know all the proteins that are capable of binding to this DNA word. Okay, a basal element, a basal DNA word that is a binding target for additional activating proteins. Some of the activating proteins are still unknown. Okay. Letter E, we have this GC element here. GC element stands for a GC-rich sequence of DNA. In other words, you would have GC, 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 you know, till you get sick of repeating it. You could have GC repeated 20, 30, 40, 50 times in a row. Okay, so this is a GC-rich DNA word. That acts as a binding target for a specific protein called SP1. Which is short for specificity protein 1. Okay. Which, when SP1 binds to the GC-rich region, has a stimulatory effect on transcription. Okay. This protein, SP1, you know, what I'm talking about here, we are really talking about things that are in current research right now. There's lots of this stuff that we know, and there's much of this stuff we still do not know, and we're learning aspects of each year. The SP1 protein is known to help regulate cell differentiation, cell growth, apoptosis, the immune response, DNA damage, and chromatin remodeling. Okay, so this guy is involved in it has its hands in a lot of different processes. Okay, let me get the last two things done, and I promise I will stop. Okay. Okay, we have here a TATA element. Okay, this is the TATA box of a typical eukaryotic promoter. And this is involved in regulating what's referred to as the basal machinery. In other words, it's the basic machinery that has to be assembled, the basic proteins that have to be assembled at every promoter of every gene in order to simply express that gene. Okay. This DNA word recruits the basic protein machinery that must be present at all eukaryotic genes in order to express the gene. Okay, that's going to include TBPs, which are short for TATA binding proteins, and it's also include TAS for TATA binding protein associated factors. Okay, don't let that bother you too much. I'm not going to beat you up with it, but this does get very involved very rapidly. Okay. All right. One more thing, then we'll close up, and that is the INR region. Okay, the letter I, INR. This is the initiator region. Okay. The initiator region is the location where transcription begins, all right? It contains the +1 site, the very first nucleotide of DNA that's converted into RNA. Okay. And why this is being put into your notes here is because this initiator region is known to be a DNA word for a special protein called PZ120. Called PZ120, which is short for a zinc finger protein of a molecular weight of 120 Daltons. Okay. So you can see where all this crazy stuff comes from. And ironically enough, when PZ120 binds to the initiator region, it actually represses expression. It turns off transcription of the gene. Okay, and I'm pointing that out because everything we put into your notes up to this point had a stimulatory effect, but PZ120 has a repressive effect. It has a repressive effect. Okay, so, okay. All right, gang. So thank you for sticking with me. This will allow me to talk about this at the start of next class and use it as a springboard for other topics in the chapter, okay? But at this point, the take-home message of a eukaryotic gene is that, wow, it's insane how many different elements the gene is involved with and has, okay? All right, so folks, do you have any questions at this time? Okay. So sorry I ran over a little bit, but I think we're at a good stopping point. So if there's no questions, I'll see you on Thursday. Remember, gang, you're in your last 2 weeks of the semester, and there's still a considerable amount of homework that has to be taken care of, right? I mean, from your syllabus that 2 chapters worth of homework is due on the day of the final exam. So there's plenty of work for you folks to do so you don't have to get bored at home, okay? So please be aware of that and try to keep up so you don't get jammed up at the very end. Okay, do you have any questions before we sign off? Okay, Morgan, let me mention to you, Morgan, I did not receive your chapter 11 homework. If you could please send that to me. Okay, folks, take care, and I will see you on Thursday. Have a good day.